Hello everyone, welcome to my channel Love of Biology. Today we are going to learn about process of recombinant DNA technology, that is construction of the rDNA, hybrid DNA. So this involves a selection of the desired gene which we want for administration into the host. So followed by a selection of the perfect vector with which the gene have to be integrated, and hence the recombinant DNA is formed. This recombinant DNA then has to be introduced into the host, and at last it has to be maintained in the host and carried forward to produce the offspring, forward to the offspring, so it will be forwarded. So this is a thing which involves the steps. Let us see those. So first, isolation of the genetic material and restriction enzyme, digestion, amplification using the PCR, ligation of DNA molecule, insertion of the recombinant DNA into host, and isolation of the recombinant cells. So one by one, let us see the steps. Isolation of genetic material, the first one. So the genetic material, that is the DNA, is isolating in the pure form, that is free from other macromolecules. Since DNA exists within the cell membrane and inside the cell wall along with other macromolecules such as RNA, polysaccharides, proteins and the lipids, it must be separated and purified, which involves enzymes, lysozymes, cellulases, chitinases, ribonucleases, proteases, etc. Other macromolecules also removed with other enzymes. Ultimately, the addition of the ethanol causes the DNA to precipitate out as fine threads. This is then spooled out to give the purified DNA. Finally, the genetic material, that is the DNA, is isolated. Next, restriction enzyme, digestion. These enzymes are the molecular scissors. Molecular scissors cut DNA at specific location. These reactions are called restriction enzyme digestions. They involve the incubation of the purified DNA with the selected restriction enzyme at conditions optimum for that specific enzyme. So if we incubate that purified DNA with these enzymes, they will cut the DNA because they will work at that particular conditions and they will cut at the specific sites. So that is a restriction enzyme digestion. So after this, we have to allow these isolated DNA into agarose gel electrophoresis, which reveals the progress of the restriction enzyme digestion. This allows us separating and cutting out the digested DNA fragments based on the size. So this electrophoresis, based on the size, we can separate the fragments. After this, ligation. Purified DNA and the vector of interest are cut with the same enzyme and they are joined together, that is the ligation, with the help of enzyme DNA ligase. The resulting DNA molecule, we call it as a hybrid of the two DNA molecule, the interest molecule and the vector. In the terminology, we call this as a recombination. Hence, this new hybrid DNA will be now going to insert into the host. In this step, the recombinant DNA is introduced into the recipient host. We call it as a transformation. Bacterial cells do not accept foreign DNA easily. Therefore, they are treated to make them competent to accept the new DNA. So, for that, there are few methods: calcium chloride induced transformation, electroporation, microinjection, gene gun method, or biolistic method. So, by using these methods, we can insert the recombinant DNA into the host organism. These methods I will explain in detail in the next video. Next, isolation of the recombinant cells. The transformation process generates a mixed population of transformed and non-transformed host cell. The selection process involves filtering the transformed host cells only. For isolation of the recombinant cell from non-recombinant cell, the marker gene of the plasmid vector is employed. So by this, we can isolate, we can separate the recombinant cells after the insertion. We can select them. Next, after this, obtaining the foreign gene products. In this method, the desired genes for administration is selected, followed by selecting a perfect vector into which the desired gene had to be integrated and recombinant DNA formed. Once this foreign DNA is multiplied and ultimately desirable protein is produced. For the production of the desirable protein, the gene encodes for it needs to be expressed. This happens only under optimized conditions. So the recombinant cells can be multiplied in large scale using a continuous culture system. Hence the cells are cultured in a large vessel and the medium is refreshed on a regular interval to maintain the optimum conditions. This helps to culture a large mass of the desired protein. This was achieved by bioreactors. So these are the large volume of the culture we can produce. The bioreactor is a large vessel where the different cells such as human or plant or animal are used to culture the new biological things. Last step is the downstream processing. It is a sequential step in which the isolation, purification, preservation of the final products are done before it is marketed. So plasmid and the desired foreign gene we take, and we have to insert it into by using the ligase enzyme into the vector, and both we form by mixing these two, we can form the foreign gene which is inserted into the host. Thank you for watching.